So the next lecture is being provided by Dr. Sharvan Sehrawat, an associate professor at the Indian Institute of Science, Education and Research, Mohali.
While Sharvan comes up, I'll briefly introduce him. So Sharvan is an associate professor in biology at ISER Mohali, and his interest is in immunology and immunopathology. He has worked on dengue, interested and is working on dengue, chikungunya, as well as some protozoal parasites. Uh, Various approaches that he uses are molecular approaches, immunological approaches, and so on. He's also interested in CD8 T cell function, and he's going to talk about immunity and immunopathology to viruses. Have you changed the title, or that's the title you will stay with?
Yeah, this is the same.
Okay, let's hear him.
First of all, thank you, Dr. Vineeta, for introduction. And I have to thank the organizers for the opportunity to give a talk on this topic, which actually I have highlighted here. Now the problem is, because whatever I'm going to talk about has already been discussed in more or less whatever we have heard today, right? But nonetheless, whatever I'm going to talk about is going to reinforce your knowledge in immunology, right? So let's start. So I'm going to show some data, but not much. Some of, rather a large amount of this data actually is unpublished. And as the title says, immunity and immunopathology. So I'm going to talk about some of the immunological concepts and end my talk talking about immunopathology caused by viruses and what kind of interventions we can actually have. Because in the end, we invariably like to rush through these slides. So, and we forget actually acknowledging people who actually did the work. I don't really do experimental work these days, except for guiding and looking at the data and suggesting experiments. Initially, I used to do those experiments, right? The majority of work I'm going to talk about today is done by Sudhakar, Surbhi, and Yuviana. And the last part of it actually was done, rather started by Manpreet. This was actually on the phase display of these antibodies that I'm going to talk about, which were selected against some of these viruses. We have collaborators who have actually provided us with a lot of reagents, and they are also helping us technically when we require their help. Dr. Stephanie Dugan from Harvard Medical School, Dr. Joginder from University of California, Davis, Dr. Karthik Chandran and Rohit from Albert Einstein College of Medicine. Dr. Rohit now actually has started his own lab, and we still remain collaborator. Dr. Ram Yadav, he's a plant biologist at ISER, and it's a unique actually collaboration because the antibodies that we are trying to generate, these are single domain antibodies, I'm going to talk about those. So we are trying to express those antibodies in plants. So at some time point, if we have to scale up the production, so that can be easily done. The funding actually is done through these different sources. Obviously, ISER Mohali is supporting big time the initial setup, and continuously every year they it's a lot of help which they provide. Ministry of Education, Department of Biotechnology, and we have a running center on antibody engineering from SERB. So these are the funding source in my lab. Okay, so a brief summary of the cells of the immune system. Just like every other cells, your immune cells also start their journey from stem cells. And after stem cells, so basically then you divide these cells into two lineages. One give rise to myeloid stem cells and another lymphoid stem cells. Myeloid stem cells give rise to the cells of the innate immune system, and lymphoid stem cells give rise to B cells, T cells, and NK cells. Now, NK cells, some people call them as innate immune cells, and some also call them as adaptive immune cells. And there was a very nice paper written by Louis Lanier, who actually showed NK cells have all the features that adaptive immune cells have. So essentially, so they recognize specific antigen, they expand, they undergo contraction, and they also generate immunological memory. So all these features actually were also shown by NK cells. However, problem with NK cells is the repertoire that you have for NK cells is limited. So this experiment or the paper that I'm referring to was with one of the herpes viruses proteins, M157, right? Now, if you talk about T cells actually come in predominantly two different flavors, helper cells and cytotoxic T cells. They get activated by recognizing the peptides. Peptides are generated through this channel that we call as proteasome, right? This antigen actually is passed through this proteasome. You generate these peptides. These peptides, if they are bigger in size, so they are going to activate your T helper cells. If they are smaller in size, they are going to activate your T cytotoxic T cells by loading onto MSC class 2 or class 1, respectively. Activated cells become effector cells, and a pool of those cells will give rise to immunological memory, right? Now, these memory cells are invariably a very limited pool, rather 5 to 10% of the cells which have expanded to begin with. B cells do not really require processing of this antigen, so they can directly interact with the antigen in unprocessed form, become activated. They make these Y-shaped structure, which we call as antibodies, right? And they also actually become memory cells, a pool of those cells. Right. Now, if you talk about types of immune defenses, let's not spend too much time into it. It has already been talked about. So essentially, you have three lines of defense: mechanical and chemical barriers, innate immune defenses, adaptive immune defenses, right? And this actually helps you fend off almost everything that you encounter with, right? Most immunologists these days actually focus on looking at B cells and T cells, right? In next few slides, I'm going to talk about the interferons, particularly the type 1 interferons produced by dendritic cells, and that too in context of aging. Okay, so this is a very trivial slide. You have your T cells. T cells have this T cell specific receptor. TCR we call it. TCR basically through TCR these cells get activated. They produce cytokines or effector molecules. B cells once activated they produce these antibodies. These antibodies are secreted product. They neutralize these pathogens. While these effector cytokines produced by cytotoxic T cells or CD4 T cells, they help actually in eradicating these infected cells. Okay, so polarization part, Amit has already discussed. I don't need to actually again dwell on this slide too much, except for saying that each of these cell types have distinct functions. For example, Th1 cells are particularly important for intercellular infections. Th17 cells for fungal and bacterial infection. Tregs are there to control inflammatory responses. T follicular helper cells for antibody production, and Th2 cells are very critical for controlling parasitic infections, right? And the role of these T cells or T helper cells actually is to generate potent cytotoxic T cell response or potent B cell response. This also actually has been talked about, where you have actually these three signals. So these APCs, they sense the presence of these microbes through these innate immune receptors, transduce signal, upregulate all these co-stimulatory molecules, B7-1, B7-2, we talked about those, right? They also produce a lot of these cytokines to begin with. The signal 3, 2, and the signal number 1, which is coming from these peptides loaded onto MSC class 1 or class 2. So once we have these 3 signals, so it basically set these lymphocytes free, and they start activating, expanding, and after that you have this clonal burst, and these effector cells control infection by killing. If these are basically cytotoxic T cells, they are going to kill infected cells, leaving behind a pool of 5 to 10% of these cells as memory cells, right? Now, if we talk about co-stimulatory molecules, so basically this is B71, B72, which interact with CD28. Now, so there is something as CTLA4, which actually is one of those inhibitory molecules which was initially discovered. Now we have actually a plethora of these molecules. Towards the end, I'll show some data which already have actually been discussed largely. But if you talk about these memory cells actually are, they also come in different flavors depending on where they are located and what do these cells express. For example, T effector cells, they are found at infected tissues or in circulation. T central memory cells, they are found in lymphoid organs, and T resident memory cells, they are particularly present at infected tissues. And these are the markers which actually can define these cells, right? Of particular interest, actually, when you think about infectious agents, are these TRMs, because these TRMs, they do not really need to recirculate back to lymphoid tissues for their reactivation when you get secondary infection. So they can actually be activated at in situ, right? So essentially that means these cells actually are able to provide very, very quick protection at the site of infection, right? So a lot of efforts are made actually to expand these resident memory T cells, right? There are a lot of strategies are actually adopted how you can actually boost the response of these TRMs. Okay, so let's talk a little bit about the interferons, right? Okay, so when we talk about viral infection, you cannot really discuss viral infections without interferons. Interferons actually are the first responder, the first molecules that are induced following any viral infection. First of all, you get induction of this type 1 interferon response. Can be produced by infected cells, dendritic cells, and plasmacytoid dendritic cells produce a lot of it, right? After that, you have all the cells. For example, NK cells are activated, then you have your cytotoxic T cells, and then your antibody producing B cells, right? And when you expand, you start expanding your cytotoxic T cells, then you start controlling the virus buildup, right? Now, this actually is a data slide. This is not actually some slide that I have actually collected from some source. So this is actually the RNA-seq data that we collected from young animals and aged animals. These young animals actually were 8 weeks old, and aged animals were 108 weeks old. And if you extrapolate this data to humans, so this is like 20 to 25 years of age, and this is like 80 to 85 years of age, right? So you collected the dendritic cells from these animals, from lymphoid tissues. You performed RNA-seq analysis, and you look at how many are significantly differentiated, differentially expressed genes, right? And one of these things that you actually clearly see here is the genes which are part of this type 1 interferon pathway. So all these are downregulated. And remember, these are not manipulated mice or manipulated cells. This is coming from naive, uninfected, unmanipulated animals. At basal level, so they have really inhibited type 1 interferon response, right? So, so what does that mean? So following aging, not only the frequencies of your T cells and B cells is compromised because of thymic atrophy and the production of your T cells and B cells is compromised, but the ability of your antigen presenting cells, your innate immune cells is also compromised, right? And if you talk about virus infection, so particularly interferon response, type 1 interferon response is compromised in aged individuals. And this actually is also, this was also highlighted by Dr. Vineeta, that aged individuals actually show huge mortality. The rate of mortality following COVID-19 in aged individual is much higher than the younger population, right? So 80 plus year, you have around 15% mortality, right? Up to 40, so this is like 0.4. Up to 50, this is 1.3%. So there is actually quite significant correlation of aging with mortality, and we have data to show that type 1 interferon response is highly compromised in aged individuals, the cells of the aged individual that produce type 1 interferon. Okay, so that was basically the RNA-seq data from unmanipulated cells. Then what we did was, so we isolated cells, PBMCs actually, from normal, healthy individuals. 6 of those from young group and 6 from aged group. And young were actually about 25 years of age, and aged were actually 65 and above. This was not even 80. Okay, and if you look at all these interferons, so basically we are comparing young versus aged, and these PBMCs were actually pulsed with influenza A virus with 0.5 MOI. MOI actually is multiplicity of infection. So essentially what that means, we are giving 0.5, rather for 2 cells we are giving 1 virus particle. Okay, so as you can visualize, that young animals were able to mount, the cells from young individuals actually were able to produce good interferon response. The downstream transcription factor STAT1 and STAT2, the tetra-tri-octapeptide, which are actually having direct antiviral effects, oligoadenylate synthetase. So all these actually were highly expressed by the cells of young individuals, but aged individuals' cells were not able to mount that response, right? So inherently, so you have problems with cells of the aged individuals. Okay, so well, you are limited by number of the samples that you can isolate from aged or young humans. So you have to do a lot of experiments with mouse. And good thing is that we have a good animal facility where we can actually maintain these animals for a long, long period of time. So now the animal facility is actually almost 5 years old. So as we have these retired breeders, so we don't sacrifice them. So we actually keep them going. So essentially we were able to actually maintain a colony of aged C57 black 6 animals, and we also have young animals, right? So we compared all these responses from aged and young animals. And when you have animals, so you can isolate all kind of cells, right? Okay, so I'm showing data from young and aged animal, but apart from that, the data actually is also shown on reconstitution of this molecule, RAB8A. Why RAB8A? RAB8A is a small GTPase which actually act downstream to these growth factors. And these growth factors actually are essential for differentiation of your dendritic cells, right? Once you have your bone marrow cells, you actually put them with GM-CSF and IL-4, and then you can differentiate into BMDCs, right? So these cytokines actually act through these receptors. Okay, so, and once you have signaling through these receptors, PI3K, AMPK, and mTOR pathway actually, which actually is nutrient sensor in the cells. So they are engaged, right? But upstream to this, so this molecule comes up. And from our RNA-seq data, we did find downregulation of RAB8A in the cells of aged individuals, right? So what we did was, I'm not showing data from young animals which were depleted of this RAB8A. But what I'm showing here is we reconstituted aged dendritic cells of RAB8A, right? And as you can see, the experiment plan actually is like this. So you have bone marrow cells from young and aged animals. And the aged bone marrow cells were also lentivirus transduced with RAB8A. So this actually is represented as RAB8A overexpressing cells, right? This was done by repeated transduction. Then you differentiate these cells and analyze for cytokine production or looking at their antigen presentation ability. Okay, so young animals, they mount actually a very good type 1 interferon response. Aged cells are quite significantly reduced. Type 1 interferon beta also shown the same thing. Gamma, same thing. But when you reconstitute RAB8A, so the expression of type 1 interferon as well as interferon gamma is upregulated. Same is the case with STAT1 and STAT2. Obviously, this is a control to show to what extent you were able to reconstitute RAB8A. Then you also have the expression of BCL2. BCL2 is a survival factor, right? Anti-apoptotic molecule. So somehow the cells which were reconstituted of RAB8A, they started showing high expression of BCL2. So that means they were actually surviving longer. Okay, so then we actually look at the antigen presentation by these dendritic cells. The experiment is simple. So you have these DCs from 3 different conditions, from aged individual, from young individuals, and the cells from aged individual which were reconstituted of RAB8A, right? And then you pulse these cells with chicken ovalbumin. This is a protein antigen, right? After pulsation, obviously, if you are adding this protein antigen, so these cells are going to take up this antigen and are going to generate class 1 peptide, which will be loaded onto MSC class 1 through antigen cross presentation pathway, right? And once that is done, now if you co-culture these cells with T cells, which have a TCR, which is specific for that peptide, so you are actually looking at the proliferation of those cells in the co-culture, right? So these are controls. When peptide was not, the protein was not added, so you don't get any response here, 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 right? Young cells pulse with OVA, so you get a lot of proliferation of your OT1 cells. Now, for students, this is a CFSE-based proliferation assay. So what essentially you do is, you have your T cells, you basically add CFSE to your T cells. CFSE is a green fluorescent dye, right? Carboxyfluorescein. This is a cell permeant dye. When you expose your cells to this dye, so this is internalized, and it basically is cytoplasmic esterases act on it, right? And then it start making adducts with free amine in the cytosol. So essentially, the whole cytosol is actually labeled, right? Now, when the cell is dividing, with every division, whatever the content of CFSE you have, that will get halved, right? So this cell is the original cell, undivided cell. When you have one round of divisions, the content of CFSE is halved. With another round of division, so this get 1/4, and then 1/8, right? And these peaks actually represent, these are undivided cells. This is 1-time division, 3-time division, 4-time, and so on and so forth, right? So more time or division means these cells actually have proliferated those many times, right? And you can clearly see that these cells, the dendritic cells from aged individuals, even after antigen pulsing, were not able to stimulate your APC, your D cells, right? Cells from young animals were able to extensively proliferate your T cells. And what T cells we are using? So these are basically TCR transgenic cells. So they have specific TCR which can recognize this synthetic peptide coming from the protein that we added. This is basically chicken ovalbumin, right? Now, if we reconstitute RAB8A to these dendritic cells, this almost recapitulated a phenotype which you get for young animals. So just by restoration of RAB8A in dendritic cells of aged individuals, you are able to induce a very potent antigen presentation and effectively activation of your T cells. Okay, so then this is in vitro analysis. Basically, you are culturing these cells in plastic plates, right? So this experiment actually was done in vivo. Now, what I'm actually getting towards is, can you somehow reconstitute immune responses in aged individuals, right? Okay, so the experiment is as follows. So we are having these congenic animals, right? We are transferring cells, and whatever the cells we are transferring, we can actually find out that these are the donor cells, and these animals, because these are having CD45.1 positive cells, right? So based on 0.1 and 0.2 staining, so basically you have antibody for CD45.1 and 0.2, so you can find out that the cells which you have put in these animals are the one that you are adding from outside, right? And you are looking at the fate of those cells. Okay, so we actually transferred 1 million OT1 cells, which were actually 0.2. Then we actually transferred these DCs from these different conditions, right? Conditions were aged dendritic cells, young dendritic cells, and aged dendritic cells reconstituted of RAB8A, right? And they were obviously pulsed with the antigen. And on day 6, you analyze these cells. The data actually is shown here. And these OT1 cells obviously were CFSE labeled to actually measure to what extent they have proliferated. Okay, so as you can clearly see that young animals obviously were inducing 60% of these cells as divided. RAB8A reconstituted dendritic cells induced proliferation of a lot of these cells. Aged were compromised. And then you are actually looking at whether these cells that you have now induced are protective at all, whether they can control this virus infection, right? And so that means now this virus that you are going to use has to have that epitope against which these T cells were generated. And for that we use this influenza virus which was encoding synthetic epitope, right? You infect these animals after 1 month with the 200 plaque forming unit through intranasal route, right? And then you are measuring response in mediastinal lymph node. This is the lymph node which actually generate a lot of immune response. Intranasal means basically the major response will be generated in a lymph node, and that is the lymph node you are sampling, right? In spleen, and as well as whatever the responses you are getting actually in lungs. In every single organ, so the RAB8A constituted dendritic cells were able to induce a lot of proliferation of, or a lot of expansion of these antigen-specific cells. And these expanded cells were able to control this infection quite significantly, right? How do you measure that infection? You take out lung samples, you actually triturate, make homogenates, and you perform plaque-forming assays, right? And you can actually count how many particles of the virus are present, right? And these are basically the lung tissues, how they look morphologically. These are young dendritic cells, old, so lung are in these old individuals. Basically individuals who are given old dendritic cells, right, are quite messed up. And here you have the rehabilitated reconstituted dendritic cells. You can actually recover quite significantly. The pathology is reduced. Okay, so how much more time? 5 minutes. Okay, good. So then I don't have to really go through a lot of other data. So this approach, see, as of now, we were actually using these OT1 cells for doing all kind of analysis, right? You can use a tetramer technology for detecting these antigen-specific cells, and this actually is generated in this way, right? Okay, so you can actually make these tetramers by cloning and expressing the extracellular domain of your MSC class 1 heavy chain. You put a tag and you refold it in presence of beta-2 microglobulin peptide. And eventually, if you actually, because this is having this biotin acceptor peptide sequence, you can put a biotin tag here, and with streptavidin, you can tetramerize it, right? You can generate this tetramer. If you want to use the tetramer for discovering epitopes, the only thing that you have to do is you have to actually generate these tetramers in presence of this conditional ligand. And conditional ligand here is nothing but a peptide which has an artificial amino acid, right? If you expose it to 365 nanometers, so this will be cleaved. And once this is cleaved, obviously the whole complex will disintegrate, right? But you can actually restore the disintegrity of this tetramer by adding another peptide of your choice. If it's able to actually sit back here, so you will get this loaded complex or displaced tetramer. And this way, so you can actually detect, this is to show the proof of principle. And using the same approach, so we discovered a lot of epitope of this gamma herpes virus, right? Now, the benefit of this approach is you just predict epitopes. You have one tetramer generated, and in a 96-well format plate, you can find out in a matter of, let's say, 30 minutes, which peptide are immunogenic and which are not, right? Rather than generating each of these tetramers for one of those specific peptides. So this is another way to basically validate it. This is basically a reverse vaccinology approach. I'm not going to go into detail of this, but approach is same. So here you actually coat streptavidin on the surface of these plates because this is biotin-islated, right? So this whole complex is immobilized on the surface of this plate. If you expose to UV radiation, so this will be disassembled. And if you add this peptide, so this will remain, and then you use anti-beta-2 microglobulin antibody. And if you're getting a positive reaction, that means this peptide is immunogenic. I know I'm rushing through it, but I have no time, right? So with this, we actually discovered some of the epitopes of dengue virus. And this is another assay to demonstrate that actually whatever we predicted acellularly is also true using RMAS cells which actually are deficient in TAP. Okay, so let's not talk about the immunopathology to viruses approach. I'll just end up. Again, this part has also been talked about where these differentiated cells start upregulating these inhibitory molecules. And if you use blocking antibodies, either against PD-1, which is one of the inhibitory molecules, or TIM3, or a combination of these two, or you combine with other inhibitory molecule, you actually can resurrect these cells which have lost their functionality. All the very recent data also suggests that only some type of cells can be revived and not all, right? So basically this is some of the rest of the slide on this aspect. Let's not forget about this. Okay, so I'll just show you one piece of data on these antibodies. 2 minutes. Okay, so we are working with these heavy chain only antibodies, right? Using phase display approach. It's easy to work with these antibodies than the normal conventional antibodies because the antigen binding portion is a single domain, right? And once you are able to produce these antibodies, and they can actually beautifully be expressed in E. coli, they don't require disulfide bond formation, they don't require any other fancy stuff for their expression and being present in the refolded form, right? You can make bivalent, bispecific, multispecific, conjugate with enzyme. You can use them as detector antibodies. The approach goes like this. You generate the library of these antibodies on the surface of these M13 bacteriophages. The rest remains that you have to have the purified protein. You coat that protein on the surface of your plate. You probe this library against that antigen. Whatever is the specific binder, it will bind. Rest of these things can be eluted, right, or washed out, and then you express it in bacteria. So we have been able to actually generate a lot of these antibodies against host proteins. Some of these viral proteins, coronavirus against proteins like cleavage site, receptor binding domain, against dengue virus. And dengue virus specifically I want to talk about because this is against galectin-3, but I'm not going to talk about this. This whole thing actually on these antibodies. Why dengue virus? Because dengue virus is a nasty pathogen that the antibodies that you generate against one serotype do not neutralize the second serotype, right? So invariably you generate antibodies if you are infected for the first time, and if you second time get infected with the same serotype, you are good. But second time if you get infected with a different serotype, chances of you getting a severe disease are very, very high. And this process actually is called antibody-dependent enhancement of infection, right? And the culprit for this whole thing actually is the Fc region of your antibodies. Now, our antibodies do not have that. It's only the antigen binding domain. And we generated these antibodies against dengue virus. We demonstrated in vitro first that in increasing dose, these antibodies are able to actually neutralize this virus in plaque-forming unit assays at RNA level. In animal model, where we are using this interferon receptor knockout animals, which were actually infected with this virus, virus pre-incubated with these antibodies or antibody was injected afterwards. And in all these cases, rather, if we actually pre-neutralize this virus, almost every animal that you infect with this virus, neutralized with these antibodies, survive. But afterwards, the survival is increased, but animals eventually die. And remember, these are type 1 interferon knockout animals. If you can extend survival by 1 day, so that means antibodies actually are pretty potent. Okay. So I think that's about it. So this is the data on coronavirus. So this is already published. It can be looked at later. Okay, you can read this slide. I don't need to talk about this, right? Questions? I know I had to rush through many of the slides.
Sorry, and I was responsible for saying only 5 minutes, but we have been running late, so I didn't want to delay things further. Questions?
Where you show the pictures of the lungs, where you the dendritic cells get more active in, if you go to the slide, I don't know.
No, so the dendritic cells were transferred
by RAB.
In similar number. The dendritic cells, what they did was, obviously they must have produced more interferon, that's one. But that is actually nothing. So these were actually 200,000 cells were transferred. And 200,000 cells, even if they are making interferons, that will not contribute much, right? The major contribution was coming from your cytotoxic T cells. these dendritic cells actually activated a lot of cytotoxic T cells, right? And those cytotoxic T cells actually were able to kill virus-infected cells, right? And that controlled this virus buildup in the lung tissues.
Yeah, my question was a little different. That is, it's since aging is an evolving area. So is it something? I mean, it has a many spectrum. So, but apart from the immunity, antiviral immunity, is it something to do with the immune downside of the immune system? We call it, the way you projecting number one. Number two, I didn't understand when you have the small size lungs over there and the middle one. So, and the middle panel was very red colored, which has
Well, red color is because there was a lot of pathology in these lungs, right?
RBCs,
So there was lung hemorrhage, a lot of actually blood vessels were ruptured, so a lot of blood was there. And why was it? Because there was a lot of virus, and virus actually triggered a lot of immune cells there, right? And that's why a lot of bleeding was there in these lungs, right? So I did not show you the data on histopathology, and that actually looked really, really bad. So the alveolar spaces were completely filled up. The animals actually, if you look at the body weight change, that was like within let's say 10 days they were almost dead,
oh my goodness.
right? So aged animals do not survive influenza virus, even if these are wild type.
Yes, sir. I had one question that RAB8A is like an aged individual. It is not able to be present in high amount, or it is not able to work like the GTP is. So, sir, at the same time, the T cells are also in the aged individuals. T cells are also like down regulated because the thymus is also decreasing in the size. So, is it not having effect effect like for activation of T cells?
Okay, let me actually tell you. So these adaptive transfer experiments were done actually in normalized environment. So we were actually looking at the contribution of the cells that you are transferring. The cells were, the only variable was that dendritic cells were either from aged individuals or from young individuals, or the one which were reconstituted of RAB8A. Rest was normal, healthy individuals,
Okay.
young individuals.
Young individuals.
Yeah.
Okay, sir. Yes, sir.
Yeah, and the reason was because we see invariably this question will come. In an aged environment, a lot of things change. So how do you make sure that these cells actually are homing efficiently, right?
Yes.
So we were actually trying to neutralize everything possible and figure out the role of dendritic cells in order to actually activate those cytotoxic T cells. And those cytotoxic T cells are also of known specificity,
Yes.
right?
Yes. And one more question, sir. The high throughput tetramer technology. So, sir, in that the peptide which was removed and which led to its disintegration of the structure tetramer. So, sir, if that is a domain, then only it will affect the structure. So the epitope, like all epitopes are the domains essential for structure of.
Well, see, I think problem was with me because I was not able to explain it well because I did not have time. So now we have time. So essentially the issue is, so you generate a tetramer with a conditional ligand, right? Conditional ligand essentially means if you expose it to UV radiation at 365 nanometer, peptide will be cleaved.
Yes.
So now your complex will be basically having no peptide.
Yes.
When the peptide is not there, obviously this cannot remain stable.
Yes, sir.
Right. At the time of exposure to UV radiation, you add your peptide of interest. That can that you find out whether it can stabilize MSC class 1 or not. If it's able to stabilize, right, whenever you are removing, you are displacing your conditional ligand, so this will go come and bind, right? And then you will get a intact structure, which will give staining to your cells,
Yes, sir.
right? when this peptide is not immunogenic, obviously this will not stabilize the structure.
Yes, sir.
And if the structure is not stabilized, it will not stay in any cells. So that means the
Okay.
peptide was not immunogenic.
Okay, thank you, sir. Yeah.
One question. What type of disease are these? You are saying this is happening through cross-presentation.
Yeah, well, I know why you are asking this question to begin with, because you know that CD8 positive dendritic cells are the one which cross-present, right? These are not CD8 positive dendritic cells. These are conventional CD11B, 11C positive CDC2s, right? However, with the RAB8 reconstitution, if these cells were able to actually cross-present so efficiently, and this is exactly the same question reviewer had, and we actually did experiment with FLT3 induced dendritic cells. We actually depleted all the macrophages from the culture of the dendritic cells, and we depleted all other kind of cells from this culture. What we did not do was use a reporter mouse which can specifically have these DC1 cells, right? But still they are okay. This paper actually is accepted in Nature.com.
You just added, OHA protein without any modification.
Yeah, it's the whole antigen. It's the whole antigen.
Any further questions? Thank you, Sharvan. Thank you very much.
Yeah. Thank you, Dr. Vineeta.
Thank you, sir. I request Vineeta ma'am to kindly felicitate Sharvan sir. We are also grateful to Dr. Vineeta for chairing this session. I'd like to invite Dr. Sanjeev Bhattacharya to kindly felicitate the moderator.